Before we explain the process of photosynthesis, first we must understand endosymbiotic theory. Before complex life emerged, the organisms that inhabited Earth were mostly single-celled prokaryotic or eukaryotic organisms because each organism did not produce enough energy to build larger and complicated cells. These organisms produce energy through cellular respiration, which was accomplished by digesting other cells, and through photosynthesis, which was accomplished by gathering energy from the sun. The endosymbiotic theory states that a larger single-celled organism engulfed a smaller organism, but instead of digesting it, the two cells developed a symbiotic relationship. The engulfed cell produced energy for the larger cell to utilize, and in return, it got the production the larger cell offered. The increased energy this relationship produced allowed for these cells to grow more complicated and become naturally selected for, resulting in the emergence of the first plant and animal cells. Biologists base this theory on the unusual presence of DNA and ribosomes located in the mitochondrion and chloroplasts, leading them to believe that these two organelles were originally cells that were engulfed by a larger cell in the endosymbiotic theory. The mitochondrion produces energy through cellular respiration, the process of utilizing glucose and oxygen to create the high-energy molecule adenosine triphosphate, also known as ATP. The chloroplast uses photosynthesis to create glucose, which is then used to create cellulose, the material plants use to grow. The glucose produced in the chloroplast is also used as fuel for cellular respiration to take place in the mitochondria, as cellular activities still need energy to function. Today we will focus on the chloroplast and the process of photosynthesis. Photosynthesis is composed of two interdependent processes, the light reaction and the Calvin cycle. The first process that occurs is the light reaction, which takes place across the membrane of the thylakoids within the chloroplast. The light reaction's main purpose is to produce energy needed to synthesize sugars in the Calvin cycle. The light reaction begins when light energy in the form of photons from the sun are captured by pigments in photosystem II called chlorophyll. This energy excites electrons located within the photosystem. These excited electrons are then transferred across the thylakoid's membrane, creating an electrical charge that helps the protein actively transfer hydrogen ions from outside the thylakoid and the chloroplastic stroma into the thylakoid's lumen. These now low-energy electrons are sent to photosystem I, where they are re-energized by light energy. Then these electrons are sent via protein to an enzyme where electrons are bonded with hydrogen ions and NADP+ to create NADPH. The electrons used in this process are replenished through an enzymatic reaction called photolysis. Photolysis is the catabolism of water or H2O into the products of hydrogen ions, oxygen, and electrons. The constant pumping of hydrogen ions through the transfer protein into the thylakoid creates a gradient. These hydrogen ions are then funneled out of the thylakoid through an enzyme called ATP synthase, which helps anabolize phosphate molecules and ADP to create the high-energy ATP. This light reaction is very similar to the electron transfer chain occurring in cellular respiration. The main similarity between the two processes is that both of them produce ATP through ATP synthase, which is powered by a concentration gradient of hydrogen ions. The main difference between the two processes is the inputs and outputs.
The Calvin cycle is the process of turning carbon dioxide from the air into the sugar G3P, which can be then anabolized into glucose.
This process takes place outside of the thylakoid in the chloroplastic stroma.
Carbon dioxide enters from the atmosphere through a process called carbon fixation. The carbon dioxide is then combined with a 5-carbon molecule called RUBP with the help of an enzyme Rubisco. The new molecule is then split in half and we are left with a 3-carbon molecule. The energy from ATP and NADH are then used to transform the 3-carbon molecule into a G3P sugar. Every third G3P sugar is outputted, but the others with the energy from ATP are converted back into RUBP. So 3 turns of the Calvin cycle are then required to make one glucose molecule. A very similar process to the Calvin cycle is the Krebs cycle. They are both very similar because they are cyclic in nature and always regenerate certain carbon structures in their cycle. For the Krebs cycle, that's the 4-carbon molecule, oxaloacetate, that is reused, and for the Calvin cycle, it is the RUBP, a 5-carbon molecule. However, both these processes also contain a very stark difference. The purpose of the Krebs cycle is to collect electrons and hydrogen for the electron transport chain, while the purpose of the Calvin cycle is to create G3P sugars and eventually are used to make glucose. Essentially, both the inputs and outputs are reversed.
Plants are able to utilize sunlight, carbon dioxide, and water to grow and create energy in the form of glucose. The two interdependent processes of the light reaction and the Calvin cycle are both vital to the overall process of photosynthesis. The light reaction takes sunlight and water and converts them into ATP and a bunch of electrons gathered from the water molecules, which are both needed in the Calvin cycle. The Calvin cycle uses ATP and electrons in a cyclical cycle to convert carbon dioxide into the G3P sugar, which can be anabolized to create glucose.